Well, welcome everybody. The Office of Programs to Enhance Neuroscience Workforce Diversity within the National Institutes of Neurological Disorders and Stroke is very pleased to welcome you to the Open Stage webinar series. This is a targeted outreach effort we have for our diverse neuroscience community to learn more about NINDS programs and network with NIH and NINDS program staff. This month, we're very excited to bring back the NINDS staff managing the Innovation Grants to Nurture Initial Translational Efforts, or IGNITE program. Before we get started, I just have a few housekeeping items. We could go to the next. Yep. So a few meeting reminders. All attendees are muted. You won't be able to turn on your cameras. So please submit any questions you have using the Q&A function at the bottom. You can upvote questions that you definitely want answered, and you can ask questions anonymously as well. You can feel free to use the chat if you have any other comments. That is open to you as well. A reminder again that this meeting is being recorded. It'll be transcribed and archived on the NINDS website. You can also enable closed captions under live transcript at the bottom of your screen. And again, we're going to send a lot of resources, including these slides, to you after the meeting. So that will be posted on our website. We'll send you an email. And please do stay connected with us and feel free to email us if you have any questions about this event or this webinar series. So with that, I'll pass it off to Dr. Roof to get us started.
Great. Thank you so much to Open Stage for hosting us. It's a pleasure to work with you guys again. My name is Becky Roof. I manage the IGNITE program, the Innovation Grants to Nurture Initial Translational Efforts. My team includes Dr. Chardell Spriggs and Dr. Chris Conrad, who are program officers in the IGNITE program and help me hold some of these grants. Ms. Shruthi Thomas is here with us today as well, and she's our health program specialist. Ms. Ashley Givens is our program operations coordinator. We have collaborated with NCCIH on one of these funding opportunities, and Craig Hopp is the point of contact there. He's not with us today, but if you have questions about NCCIH's participation or if your project might be a fit for that, he is your point of contact. We're going to go through 3 different FOAs, and then we'll answer questions. If your question is super specific to your particular project, I suggest reaching out to us via email, and we can set up a call to discuss that. But we're happy to have questions about the program in general here in the webinar. All right, so the goal for IGNITE is to get you ready for later stage programs such as the Blueprint Neurotherapeutics program for small molecules or for biologics. This program IGNITE was specifically designed to get you the entry criteria for these programs. It's a really good idea to look at the later stage programs and make sure that your IGNITE project is going to get you to those entry criteria. We have 3 different FOAs that we'll be talking about today in detail. One for assay development and therapeutic agent identification, one for development and validation of model systems, and that includes in vivo and in vitro animal or models. One for neurotherapeutic agent characterization and efficacy studies. All 3 FOAs have the same budget, and that's $750 direct costs for the entire 3 years max, and no more than $499 direct costs in any given year. We have 2 upcoming due dates, one in October and one in February. We have a published 3rd one in June as well. I always get questions about late submissions or continuous submissions as a reviewer. Here's the policy. Take a look and let us know if you have any questions. All right, I'm going to spend a minute on the mechanism because this is not like your standard R01 or R21. We use an R61, R33 mechanism for IGNITE, and this is a 2-phase award. You can think of the first phase as you're setting things up, your early feasibility work, and then you will have a set of milestones, and then you move forward to the R33. And you can think of this as your pivotal experiment, your proof of efficacy, or your screen, or whatever it is that gets you to the next step. And why do we use a mechanism like this? We do this because it allows for dependent aims. The R33 depends on the R61 working, okay? The first phase is preparing, and then the second phase is executing, the major event. So the first phase might be setting up your assay, and then the second phase would be executing those assays in a high throughput screen, for example. Okay. The benefit of this with the dependent aims is that we can support, stepwise work. It's not exploratory work. We're moving towards a direction. We're doing early stage translation, and so we should be moving forward. Now, the reason dependent aims work in this mechanism is because of these milestones, right? So review is okay with R33 depending on R61 working because those milestones are there. That's what de-risks those dependencies. So it's really important that those milestones be really clear and be really quantitative and be really strong. Okay. The transition from the R61 to the R33 is done via an administrative review. What does that mean? That means it doesn't go back to peer review. We don't have to take that time. Review saw the milestones up front. The administrative review is internal at NIH, and that just helps us move more quickly. If your milestones are met, your paperwork's in on time, you can go from the R61 to the R33 without any gap in time. All right. There's a single go, no go decision point between the R61 and the R33. Some people will put milestones in their R33, and you can do that, but it's not a go, no go decision at that point. All right. Now, because milestones are probably the #1 hit on thing in a summary statement, it's something people have a hard time with when they're new to the mechanism, and it's something that reviewers are really good at looking at. So I'm going to walk you through some examples here. So hopefully you get the idea of what kinds of milestones are reviewers looking for. For example, a weak milestone might be we're going to scale up our compound. A stronger version of that is we need X grams, we need X purity. Okay, you can see where that's more quantitative. One thing I see sometimes people put in a milestone that says, we're going to do PK studies and we're going to look at 5 different parameters. It's going to be really comprehensive, and then we're going to pick the best compound. Well, what does best look like? What do you actually need? What would reflect that the R61 was successful and that you have something that's worth testing in the R33? Okay, the word best doesn't capture that. You could spell out, well, this is what we would need to see. We need to see a certain brain exposure, for example. And that should be justified based on what you know you need based on your compound's activity. This is just an example. A fully optimized and validated assay. So if you're doing the assay development screening FOA, this is a common milestone that you might need. But what does optimized and validated look like? A reviewer would probably comment that this milestone isn't strong enough. But you can put some metrics on that. You need a certain Z prime. You need a certain signal to noise. Okay, you should be getting the idea here. The last example is, our protein will decrease seizures or have some activity. People will commonly say something like, oh, we need to see a statistically difference between our control and our treatment. And that is quantitative, but does it reflect success? Does it mean anything that would reflect anything clinically meaningful? So reviewers probably would want some justification for what you need to see. And we have some examples on our website. You can find them here. Again, you'll have these slides, so you can click on this link and get more examples. All right, with that, I'm going to turn it over to Chris to talk about the first FOA.
Thank you so much, Becky. Yes, so this FOA is PAR 21-2124, and it's for assay development and therapeutic identification and characterization. And this helps support one of the early and important stage of assay development and follow-on activities. And the goals of this FOA are to develop new translational assays, and then these screening approaches really will help to identify and characterize novel therapeutic agents. And as Becky said before, for these particular announcements, there's entry criteria. So these need to be included in your application. So as for this FOA, you would want to have how is this approach novel, like this assay, and how is it better than what's currently existing? And then you want to have a strong biological rationale for your approach. And then what is the relevance for therapeutic development, and especially a indication that is important to the NINDS mission in this particular case. And then one of the other important criteria too is the what test agents are you going to be using for testing this assay development, or what kind of library, just to really know, and also providing a rationale for that choice, of particular agents. And here is just some examples of what you could propose for this first stage, which was brought up before, R61, and that's, can include some of the following here: development and validation of assays, including phenotypic assays or target-based. And these are part of a succinct testing funnel that help, you triage your testing agents. And then it could also, or it could be including a development of in vitro or in ex vivo potency assays, or even efficacy endpoints. And then you could also have development of assays to evaluate certain properties or points in, shown here, like in cellular processes like cellular uptake or engagement, or even downstream functions. And some of the other points that you could bring up in your approach would be assays to evaluate your purity or identity of your particular therapeutic that you're working on. And some of the more broad ideas too for this would be, if you're going to be doing assay development and optimization for a particular high-throughput screen, which is a more common approach. And in the event of, one of these approaches or activities is not enough, you might have a combination of different biological assays that maybe help you address your identification of your particular therapeutic agent for your indication, of course. And next, as Becky talked about, you're going to need to have, for that R61, what activities are you doing and what kind of endpoints or milestones are you going to have that you're going to reach that show feasibility for going on to the next stage. And we just showed some examples here, showing that, do you have good criteria for assay development optimization that shows that you completed it? And these have to be quantitative, of course. And do you have the right chemical and biophysical characterization of the test compounds that you're going to use? And also, is there any kind of further development or selection needed for your vectors? Has that all been completed for your next phase? And all these will be considered as your no-go criteria for going into the phase 33. And shown here are just some examples. Again, these are just examples of, what you might be doing in that later stage. And that could be preparing and screening a select series of therapeutic agents, like described before, like a high throughput screen or a high content screen. And the preparation of, therapeutic agents and confirmation of structure, doing that more identity work, for a particular therapeutic. And, using, different approaches like computational analysis or even physical chemical properties, just to really fine tune your therapeutic agent properties, knowing which way to go, to further develop your therapeutic. And just some early in vitro, I guess, pharmacokinetic activities you might want to consider as well. And then assessment of off-target, because you want to make sure what is the specificity and potency of your particular hit or lead, and then further optimization beyond that. And then, as not only when you apply to this RFA or for this a PAR, that you want to, be part of the NINDS mission, but then also the NCCIH, which is interested in, a different area of approach and special interest stated here, you want to focus on their mission if you're applying to that part of the announcement. So here, they're looking for assays that are very focused on analgesic assays for either medium or high throughput screens. And this has to be with natural products. And shown here, these are some examples here, whether they're natural products from extracts or different fractions, whether they're herbal or botanical or marine. They're just showing some examples. And if you have any further questions on this particular part of this announcement, you can contact Craig Hopp for that. And as with all focused announcements, there are certain activities that are a little bit outside. So you just want to make sure that you're focused on this area, because not that these are not important, but for this announcement, we want to focus on certain activities. So some of the things that are outside of this announcement that would be considered non-responsive would be anything that's focused, as I said, outside the NINDS mission or NCCIH's stated interest. And then also anything that's going to be looking at biomarkers specifically, and then also looking just specifically at the PD markers or any kind of work that is not really working toward the therapeutic. And also, since we're looking at trying to find therapeutics, we're not looking at devices here or surgical procedures. These are all very important, but we have other announcements for those at NDS. And then if you have an assay that is just going to help you with your basic research, we might not be seen responsive. You have to show that it's going to be in part of that therapeutic development. So you want to make sure you focus on that area, not just basic research or target identification or even just mechanism of disease, which is very important, but it's more of a basic science question. And then in the last points here is just on the in vivo efficacy, which is important, but that's serviced by another one of our FOAs that you'll be hearing about a little bit later. That's not really part of this support. So we want to make sure that. And then also just the last part about focused clinical studies, using this for bioassays for clinical development or IND enabling studies or human subject research. These are all areas that you'd want to really talk over with your program director before trying to apply for this FOA. But please let us know if you have any further questions at the end here, and I'll pass it off to Shruthi.
Thank you, Chris. So the next funding opportunity in IGNITE is PAR 21-123, which is the development and validation of model systems. So the goal of this funding opportunity is to promote a significant improvement in the translational relevance of animal models and ex vivo systems that will be utilized to facilitate the development of neurotherapeutics. In addition to biological rationale, translational rationale for the proposed model system or ex vivo system and evidence of value for future drug discovery and development are part of the entry criteria. And so essentially, we're looking to provide translational tools for drug discovery and development that can not only be used for your lab, but other labs and can make a difference in the field. And Becky will cover this a little later, but this funding opportunity is also responsive to a notice of special interest developed through the HEAL initiative, focused on the development and validation of pain-related models and endpoints to facilitate non-addictive analgesic discovery. All right, so there are two important concepts to define for this funding opportunity: internal and external validation. Internal validation refers to the consistency of the experimental design, including precision, reliability, analytical sensitivity, accuracy, and dynamic range characteristics of endpoints utilized to assess the animal model or model system. Whereas external validation refers to the extent to which a study can recapitulate aspects from one setting to another, such as the preclinical to clinical space. And external validation can be thought of through three components: face validity, construct validity, and predictive validity. So face validity would refer to similarity between the model and the disease, such as symptoms and morphology. Construct validity would be the similarity between your model and the physiological basis of the disorder or the disease mechanism. And predictive validity would refer to the similarity between the effect of a validated therapeutic intervention in the model or model system and the disease population. So Some examples of activities for the R61 phase include initial development of the model or ex vivo system. So you can develop a brand new model, or you can use a model that has not been well validated and validated. Any activities that include optimization related to feasibility, endpoint range, sensitivity, internal validation for endpoints used, and scale up for the R33 phase. So for example, if you are using a genetically modified model, you would want to make sure that breeding is sufficient and that you have stable generation for the R33 phase. Examples of activities for the R33 phase include all external validation studies, including comparisons of phenotype to human disease, comparisons of disease etiology and preclinical species to what is known about the human disease and efficacy of clinically validated therapeutic agents. And I'd like to emphasize the last point because there are many neurological disorders that do not yet have validated therapeutics. So it's not necessarily expected that you can provide predictive validity for your model, but overall you should still emphasize a rigorous approach that is translational. And so the model should mimic the human condition as close as possible, and the endpoints being used in your preclinical model should be the same or similar to the endpoints being used in the clinical setting. So in this NOFO, there are also non-responsive studies, and those would include development of models for the purpose of understanding disease etiology and identification of CNS drug targets, discovery and development of biomarkers and pharmacodynamic biomarkers. If you do have research interests here, they may likely be covered in the NINDS biomarker funding opportunity space. And also non-responsive are studies that are covered by the other NOFOs mentioned here today, including screening, efficacy, and PK of your therapeutic agent. Also non-responsive is discovery or development of devices, device-drug combinations, surgical procedures, diagnostics, or rehabilitation strategies, and human subjects research that does not meet the exemption 4 criteria. All right, and I think next is Becky.
Hey, great. Thanks, Shruthi. So the third and final NOFO that we're going to talk about today is PAR 21122, which is Neurotherapeutic Agent Characterization and In Vivo Efficacy Studies. This is our most popular NOFO. The goal here is to demonstrate that your early stage therapeutic, biologic or small molecule has sufficient biological activity to warrant going to the next steps, such as a blueprint project. And we support PK work and PD target engagement work, as well as in vivo efficacy studies. The entry criteria here are that it's novel, and that there's biological rationale, and that there's relevance for therapy development. So some examples of things that we would support in the R61, I'm not going to read this all to you because you'll get these slides, but for example, pharmacokinetic work or distribution work, studies to confirm selectivity, development of your target engagement pharmacodynamic marker, any other preparation that you need to do for your therapeutic agent or to characterize your agent. By the end of the R61, it's hoped that you'll have your agent fully characterized and ready to go. You've got your PK work done, so that informs your efficacy work, and that everything has been done rigorously. In the R33, the main activity people usually use this for is the in vivo efficacy work. We also support pharmacodynamic work, PK-PD relationships or PD efficacy relationships, replication studies. Really preliminary tox is also allowed, although pay attention because IND enabling tox, GLP tox, that's not allowed. That would be a later stage blueprint project. All right, so things that are non-responsive, again, just like the last two, is things that would be either basic science that's better served in an R01, later stage stuff that's better served in a blueprint project, or things that are better served by other programs such as the biomarker program, the device program, or the other IGNITE NOFOs. So as well as human subjects research. And I will mention really quick, people are sometimes surprised to find out that human subjects research can include work done on human tissues or samples, depending on whether they're de-identified or prospectively gathered or what. Just read the NIH guidance on that carefully. Okay, and I do want to mention there's some special features here. So we have some NOCs, which are notices of special interest. And that basically is a thing where NIH puts out and says, hey, you know, we would really like to see applications come through an existing PAR. We have one asking for animal models of pain that would be supported through the HEAL program. We have one for ALS work that would point to any of our IGNITE FOAs. If you're working in this space, I really encourage you to apply to IGNITE through the notice. And that allows us to use special set aside funds for HEAL or ALS to fund your work, which is to your benefit whenever you can use a set aside. We have a separate RFA for early stage therapy development for Alzheimer's disease related dementia. It's very much in the same space as IGNITE, but it's its own separate RFA. This due date is really soon. So I hope if this is something you're interested in, this is doable for you, but happy to talk about that as well. And we have lastly a special ADRD FOA for models that would reflect ADRDs, as particularly multiple aspects of disease. And so that is actually has the same due date as IGNITE. And that's also something to consider if you're in that space. All right, now I want to give you some general tips before we move into the Q&A. And this is just based on things that I've seen over and over again in review or you know, things that people commonly ask about. So first of all, contact us in advance. I know it's a lot of work to put these applications together, and you don't want to do all of that and then get here and find out, well, it's not quite a fit for the FOA, or you missed a non-responsive criteria, or your NINDS doesn't support that kind of research. You don't want to do that, and we can help you make sure that you're in the right place before you do all the work of an application. NINDS has been talking about rigor for a really long time, so it should be no surprise to anybody that we expect rigorous studies, design, and supporting data. This is particularly important for translational work because you are hopefully setting the stage for bigger, more expensive later work and eventually work in human subjects. So we really want all of that on a really strong foundation. It's a good idea to have a multidisciplinary team. I will point out that the reviews here are multidisciplinary. We haven't talked about reviews yet, so I'm going to take a minute. This is not reviewed by CSR. It is reviewed by NINDS using panels set up by NINDS SROs. And these reviews are very multidisciplinary and very translationally focused. Okay, it's not your basic science exploratory review group. And so you'll have reviewers who know the disease, but also who are experts in therapy development. You'll have clinicians, you'll have industry people, you'll have academics on the panel. And so you will have a lot of different perspectives looking at your application. So this is your audience. The best way to write an application that appeals to such a diverse audience is to have a multidisciplinary team yourself who's helping you write it with all of these different perspectives in mind. Let's see, next, NIH is striving to increase the diversity of the workforce. And you can help us with that goal by increasing the diversity of your team. Discuss intellectual property. So this is something that is unique to IGNITE compared to like an R01. You will have the opportunity to add an attachment to your application where you can talk about intellectual property. And so I often get asked, how detailed does that need to be? You don't have to have patents filed, but you should have a plan. You should convince reviewers that this has a path and that there's no existing intellectual property that blocks you from doing what you want to do in the IGNITE program, and that you're thinking about the next steps. The last thing you want is for reviewers to question whether there's a path forward, even if this is successful. Discuss your therapy development plan. So that's also something that's unique here compared to like an R01. And again, it doesn't have to be super detailed, but you should convince reviewers and NIH that you're thinking about what the next steps are. Once you're successful in IGNITE, then what? Where are you going? What's your next step? What's your patient population? Okay, are there barriers that you're going to encounter in the next steps that you're preparing for and you have the expertise for? That kind of thing. Just, again, convince reviewers you're thinking about it. An administrative note is that we will be requesting, you'll be needing to request approval before any no-cost extensions. And that's just so that we can track it better and better understand why we're getting NCEs. NIH has a new data sharing policy. This has been around for a little while. Hopefully this isn't new to you, but please carefully take a look at it. There are entire webinars on this subject, so I'm not going to spend time here, but if you have questions about how it applies to your situation, please reach out. All right. If you're SBIR eligible, I encourage you to use that program instead, because again, there's a set aside and you can potentially benefit from using the money that NIH sets aside for small businesses. We've already talked about milestones, but they need to be there. You need to clearly demarcate what activities are R61 versus R33. That should make sense based on what we talked about with the phases. They should not overlap in time. Pay attention to non-responsive activities and pay attention to human subjects. Everything with an asterisk here is a non-responsive issue. So please read carefully and please ask us if you're unsure. All right. And then one last slide before the Q&A. Resubmissions, most applications don't get funded. This is true with IGNITE as well as any other NIH funding opportunity. Sometimes it's a really good idea to resubmit. Resubmissions have the same due date as new applications. So that's something to pay attention to. Statistically speaking, in general, resubmissions tend to score better. And so don't shy away from resubmitting even if your score isn't very good, because I think there's some people feel like, oh, reviewers didn't like it, let me start with a clean slate. Well, that's not always the best strategy. But pay attention to what reviewers said and take it to heart and use that to improve your application. And we're happy to talk about it. So once you get your summary statement, please reach out and we're happy to set up a call. All right. So with that, thank you so much for your attention, and we're going to open it up to the Q&A.
I think we have one question. Someone asked, can someone apply directly to the R33 phase if R61 studies are completed?
No, you can't. You need to at least do something in the R61, but I'm happy to talk about strategies there if you want to reach out with your particular aims. Any other questions? All right, well, while we're waiting for those to come in, I'm trying to remember any questions we had last time we did this, and I can get us going. One question we had last time, I think, was whether or not foreign applications are allowed, and the answer is yes. No, I'm sorry, I'm trying to also read the chat. Sorry, we should have had some in our back pocket. This did not happen last time.
We have one through the Q&A. What portion of funded applications are academic versus business?
Ooh, that's a good question. So we typically send the small businesses to the SBIR program because then they can use the SBIR set aside. And so we don't have a whole lot in IGNITE. It's not zero though, but most of them would be academics just because of, encourage the SBIR program.
Someone also asked, how many applications do we normally get in a cycle?
20 to 30 applications per cycle.
Is there an expectation that these awards will lead to direct human clinical trials?
No, if you're almost ready for a clinical trial, I would send you to the Blueprint program. So this should be earlier stage work. The Blueprint project or program will support further optimization, scale up SAR work, PK work, IND enabling work, all the way through a first in human clinical trial. And those are 5-year grants. So if you're thinking that you're almost ready within the next few years, then Ignite is maybe too early for where you're at. Okay.
We have one, shall omics studies, proteomics, SN, RNA-seq, et cetera, be part of the internal validation or external one?
Okay, so I guess this is in reference to the model FOA or NOFO. I think it kind of, I would think that's perhaps internal validation, but I think it kind of depends on the bigger picture and what all your goal is and why you're doing those studies. So I'd encourage you to reach out, send us a names page, and we'll be able to set up a call. And I guess since we don't have any other questions, I'll also say, you know, I've said a couple times, reach out to us. It's typically easiest for us to help you if you also send a names page, and then we can make sure we have the right people on the call and that you're, you know, talking to the right folks when we set that up.
We have another question about milestones. If year 2 and year 3 of the R30 are for the R33 phase, do we need to have milestones for each year?
No, there's only one milestone transition between R61 and the R33. If your R33 is 2 years, spans 2 years, then in between would just be a regular progress report. Like I said, some people will put milestones in their R33, but then it's not a go, no go milestone. It's just a vehicle to tell reviewers what you're hoping to achieve. All right.
One more. We have, what advice do you have for early stage biotechs?
So if you're a biotech, again, go to the SBR program. If you're eligible, they actually have a whole bunch of programs to help, not just with your grant, but they have programs to help you get off the ground and get talking to folks, licensing or whatever you need. There's a whole big program on that I don't know all the details of, but if you're working with them, they can hook you up with some of their resources.
Do you have examples or resources for milestone development? Yes, we do on the Ignite website. They are linked throughout the PowerPoint and we'll send those out to you. With regards to clinical data, do we need to redo the assays using patient samples, or can the data be mined from existing published studies and compared to our models?
Okay, so there's a lot of context missing from that. I'm going to guess that you're talking about, because you're talking about models, that you're talking about the model development FOA, and you're trying to understand how the model that you're developing reflects the human condition. So you actually cannot use human subject research in the model FOA. So depending on where your patient samples are, like that may or may not fit. I'm sorry, I'm rereading this. So I guess it gets down to, does the data need to be done in our hands, or can we reference existing data in terms of entry criteria, entry feasibility? I think that's where you're getting at. In general, you can have preliminary data that's either in your hands or referenced through literature. If it's not in your hands and it's necessary that you are able to do it for the future work, you know, then you just got to demonstrate that's feasible. I'm sorry, I'm missing a lot of context here, so I'm just trying to guess at where we're going. But reach out to me with the names page and we can talk about the details. If that didn't answer your question, I'm sorry. Any other questions? We're just going to give it one more second to see if anything comes up. Going once. All right. In that case, if something occurs to you later, feel free to reach out. Happy to talk about any particular project plans or particular aims page. Again, you can send a names page to me and we'll be happy to set up a call. And with that, I turn it back over to Anna. Thank you.
Thank you all so much. Thank you for attending today. I know it was a lot of information, so hopefully you can take this time to absorb it. We'll send you all the resources afterwards. And part of this webinar is just to see the people that are behind NINDS and the people that you can ask questions of. We are public servants here to help you. So please do take advantage of these opportunities to learn more about future programs and what we've got going on. Please do stay connected with us. We have a great open stage coming up next with our Hispanic and Latinx colleagues. And I think I see one more other question in the chat. Just to say, we'll be sure to send the recordings and the PowerPoint slide and everything to you afterwards. And if you do want to stay connected for all other future diversity-related training and NINDS resources, please do subscribe to our listserv. With that, I'll close this session and say thank you so much again to our panelists and attendees for joining today. Bye.
And thank you to Open for hosting us. Thank you so much.
You're welcome. See you next time.